So now we're going to talk about the plant pathogen, or the plant pathogen and the interaction with the disease triangle. Pathogens are living organisms like fungi, bacteria, virus, and nematodes, and they're infectious. Okay, so they're infectious means that the organism is capable of penetrating a living host and then subsequently developing within the cells and using those cells for its own nutrition. It's also transmissible oftentimes, so you can take it, that pathogen from that infected plant and then transfer it to another plant. So it's transferable. Abiotic factors, I'm not going to spend a lot of time on this, but these are the non-living factors causing physiological disorders. Oftentimes, some plant pathologists don't like to call them diseases. They only relate to living things like bacteria or fungi that cause disease. So sometimes they're called a disorder, but we're going to call it a disease because it fits into the disease triangle very nicely. But drought could be a abiotic factor. Frost, nutrient deficiencies, excessive salts, herbicides, even air pollution can be considered abiotic factors that can cause, malfunctions of the plant, which is really what, which really is a disease. And these are non-infectious.
Too much water also.
Too much water, big time. In the case of too much water, let's say in a pot, you're filling up all those the air spaces, and it's excluding the oxygen. And what do roots need? Roots need oxygen. So in this case, the abiotic factor is exclusion of oxygen. It's too much water or too much watering. But the process is that you exclude the oxygen. The roots need the oxygen for respiration, and then they start to suffer, and then pretty soon the top starts to suffer. Okay, these are non-infectious, of course, because they're not able to infect and develop like a living thing, of course. I think you've seen these type of diagrams where in this case, corn, various mineral nutrients are needed for certain physiological functions in the plant. And if a particular mineral nutrient is required for a particular function, then you'll see that the manifestation because of that process not happening in the plant. For example, iron is very important for the chlorophyll molecule, chlorophyll, the green pigment in the plant. So if you don't give the plant iron, you don't have that ability for that plant to produce chlorophyll molecule, and you'll get various manifestations of the disease or the disorder as in chlorosis, the lack of chlorophyll. So, let's see if there was anything I have to...
And well, you looked at that, and we have another ABCs just for nutrient deficiencies or nutrient management. So we talk a lot about how to identify the deficiencies or the toxicities.
Yeah.
So you guys, that's what you are dealing with instead of just the pathogens. Feel free to attend one of those.
I think the only thing I wanted to add to this is that one of the things that I see oftentimes is the excess of salts. In other words, you're not leaching a pot out enough, and those salts, those mineral salts that naturally build up, are increased. And you get stunting in the plant, but you don't at first. But you don't and you don't know you have it unless you have something that is leached properly or whatever. Or you might see it if it's a seedling the seedling just can't tolerate higher salts. But salts, I think, is one of the real common kind of under the radar problems.
Especially in container production.
So that's a whole other class. Abiotic factors, air pollutants is a good example. Ozone, these are examples of ozone which is produced in automobile exhaust. Really, our orchids are super sensitive to that. And in this case, selenium fern. Okay, other air pollutants, nitrogen dioxide, sulfur dioxide, and ethylene is one that's quite interesting. Every so often, if you have incomplete combustion in a greenhouse, a heater in the greenhouse is not vented properly, so with incomplete combustion, you get ethylene production. Ethylene is one of the 5 major plant hormones, and you'll get a very strong reaction to a lot of plants like orchids, and you get flower drop, malformation of vegetative parts off of. That's another abiotic factor. Okay, so the pathogens that we're going to be talking about: fungi, bacteria, viruses, and nematodes. I'll just kind of give a brief overview. Fungi are multicellular organisms that grow in filaments, called hyphae, so almost like threads, microscopic or just above a microscopic level. They collectively, all those hyphae are called mycelium, and they have the ability to secrete like enzymes in their host, so they're able to absorb the nutrition. So they're really parasites. Really, they need that plant to absorb nutrients that they need from the plant. The cell walls are not like plant cell walls. Plant cell walls have cellulose in them, but the fungi have hyphae, which is the same material that's in the exoskeleton of insects. So these guys use hyphae. Fungi reproduce by various spores, and as we go through this, you're going to see lots of different types of spores: metaspores, zoospores, orangia, conidia, etc. And bacteria are tiny single-celled organisms, so they have a cell wall on them. If they have a thick cell wall, oftentimes it can be strained. It can be cultured. They can be stained with a gram stain, gram-positive. There's a lot of gram-positive human pathogens. Actually, most of our plant pathogens are gram-negative. They have thinner walls, and the gram stain doesn't stain, so they're called gram-negative. But there's other types of stains that will actually stain the cell wall of bacteria. The bacteria have a flagella on them. They're different shapes, so they can be spiral shaped, they can be round, they can be rod shaped. Most of the plant pathogens are rod shaped. They have these flagella. The type of orientation of the flagella can be an identifying feature. Some like Fusarium or Erwinia have flagella all around them. Others like Xanthomonas are called polar because there's flagella all around the inside. And the flagella are used to propel the bacteria for short distances in liquid or water. Just short distances, they don't go very far. We're going to have an example of bacteria.
No, we are not going to have.
We're not going to have it. Well, we'll see something.
Yeah.
We have lots of bacteria over there. Whether you'll see it in a light microscope is up to
We can see them in the dissect
see them. And these are, some people don't even say they're living, you know. There's a controversy, but we should probably call them living. But, and that's because they only have a little bit of, they have a genetic part to them, either RNA or DNA, you've heard of that, and then they surround themselves with a protein coat. But they need, I think why they say that they're not even living is because they need really another host to reproduce. So they sort of take over the machinery, the physiology, the genetic machinery of their host, and then they can reproduce. And so this taking over is part of this manifestation of their symptoms. Nematodes are non-segmented roundworms. Some of them takes a microscope to see. Some of them, especially the female root knot nematodes, you might have seen that on roots. They're very you've seen them readily on the roots causing these root nodules. So there's a really interesting feature that I saw, or fact, I should say. 10% of, okay, so nematodes are the most abundant multicellular animal in the world. About 10% are obligate parasites or biotrophs that are plant pathogens.
And they,
or it can be human pathogens also. Yeah.
and the most abundant animal.
The most abundant animal.
Is that by biomass or by species?
By numbers. And also they distinguish between the non-parasitic and the parasitic by the style.
Yeah, okay.
That's a very key characteristic.
We're gonna show that with the stylet. Yeah, the plant parasitic nematodes have a stylet. Now, just to give an idea of shapes and sizes of the pathogens relative to a plant cell through this, and this is one single plant cell. This is the cell wall. This is the nucleus of the cell. This gives you an idea that fungi are pretty large. They have this multicellular. A bacterium here's much smaller. There's the flagella. There is a group of bacterium that don't have cell walls, and they're kind of different shapes. Those are called phytoplasm. And then viruses, I don't even know if I should. I actually added that dot just so that I could show you something. But
Smaller than that.
It's probably smaller than that because it requires an electron microscope to see.
And then the nematode, that's the head.
Oh, wait, okay, there we go. There's the nematode. And guess what? There's Maria's stylet there, which we're going to see also. Okay, so now here's another concept in plant pathology, is that pathogens can be biotrophs or obligate parasites, meaning obligate or obliged, I guess, to be a parasite. These are fungi that have very specific host ranges. For example, with rust, this rust fungus only attacks euphorbia. This one only attacks penstemon. This one only attacks poplar. The poplar rust pathogen does not attack penstemon or euphorbia, for example, and vice versa.
And in practical terms, what it means is that you cannot cultivate them in like a nutrient agar to identify them. So you have to reproduce them if you want to study them only on living tissue.
These are not, they don't grow as saprophytes, so that's another term, meaning something that can grow on dead or dying matter, or for that matter, potato dextrose agar in a culture plate. And so that's why you have to have they have to be seen on a living host. So if you need something, if you want to send this to the lab to be, quote, cultured, they're not going to culture this. What a lab is going to do is to try to get the spores to come out on the living host. And it's the spores, it's the structure of the spores that is used by a plant pathologist to identify a particular pathogen. And the host that it's on, of course. The other kind of extreme is called the necrotrophs. So these are the ones that have this ability to very well colonize dead organic matter. Many of these are facultative parasites or saprophytes, meaning facultative, meaning they have the ability to switch back and forth depending upon the situation. So, botrytis is an absolute best example of this. It's certainly one of the most important pathogens that we have in greenhouse, nursery, culture. Botrytis, these are different examples of botrytis here. But here's an example where a pathogen, potential pathogen, can be colonizing the lower portions where these leaves are laying down there and you have dead leaves, so it's colonizing the dead leaves, and it's basically just waiting to blow up for the right environmental conditions, and then it can be spread by aerial movement through the greenhouse and land, let's say, on a flower or actually on an injured or wounded branch. And that's what I think occurred here. This is in a greenhouse that I saw where it was colonizing this dead leaves and then moved up onto a cut stem. So they were harvesting the Limonium flower here. They left the stub and jumped from those dead leaves onto the dead stub and then moved back in. And what I didn't show here are all the dead plants also, because after the botrytis got going there, it was able to move back that dead stem into the crown of the plant, and it was killing the plant off. I took this picture just last fall, poinsettia time. What was happening here is that the grower was leaving a little stub, he was doing some pruning, left a little stub here, and the botrytis was colonizing the stub, moving back into the main stem and girdling, cutting off the rest of the stem. He had this major problem here because of these little stubs that he had left on there and was not protecting those stubs in the wounded tissue with a proper fungicide. Here we have flowers that were infected with botrytis, placed in a bunch of good flowers, wrapped up and real tight, and when we got to the florist or whatever, to the wholesaler, and they were opened up, guess what? This was the one dead flower turned into many dead flowers and a clump of botrytis. Oh, what I didn't mention, but we mentioned it later, gray mold is the common name for obvious reasons because it's Canadian gray. And you know what? I'm going too slow because I think I'm having too much fun now.
It's reminiscing of old
yeah, when I see, I have, so that's why I just put this together, because I see these and I remember these situations in the nursery that I hope that I can convey some real practical knowledge on how to control them. Anyway, fungi and bacteria are spread by various conditions. Water splashing, wind and wind-blown rain, and by the way, there are particular structures, spray and runoff, so organisms can move in the runoff, contaminated seed, buying disease-free seed, make sure it's from a good source, infected transplants, soil and debris on the bottom of shoes, Make sure you remove all the soil from the bottom of the chute before you go into a new area. Tools and equipment, again, mainly remove the soil from the stools. Infected plant debris left in the garden. So, you know, you have your favorite plant in the nursery sitting over there in the greenhouse in a pot. Oh yeah, that's the tomato plant because everyone likes tomatoes, right? So we're providing tomatoes. And that sitting over there has a tomato necrotic ring spot. And it's being, the thrips are transmitting over into your snapdragons or something. So anyway, be careful about those favorite little hobby plants that are inside. The other kind of concept of plant disease, plant pathology is a disease cycle. In this case, the example is fire blight caused by this bacterium, Erwinia amlivora. So in many of these diseases that we'll be discussing, you can come up with a life cycle. And I know David has placed out some life cycles for many of these organisms out there, and you'll see them printed out. And so this is the part that I was telling you about, that if you can understand this, you try to break the life cycle here if you understand this. The first thing, identify the plant pathogen. Second, look up in some references or online, UCIPM, discover what the life cycle is, and then figure out what you're going to break here, what part of the life cycle you're going to break. That's how you manage diseases. Okay, now the concept of a sign of a pathogen. The sign is this visible, what you can see with the naked eye, what you can see with a 10x hand lens, what you can see in a stereo microscope, what you can see in a light microscope, what you can see in an electron microscope. The actual pathogen. And if we can see the actual pathogen, as I mentioned, the structure of it, whether it's multicellular, singular cellular, whether it has flagella on one side or the other, the shape of the bacterium, virus or not, these plant pathologists, or any plant pathologist, or maybe even you, can help. It all helps to identify the plant pathogen.
I should add that it's the presence of the pathogen or a part or product of the pathogen.
Yeah.
Like in the bacterial ooze, you're only seeing the capsule.
Okay. So what Maria's talking about, like, for example, when you see Xanthomonas or Pseudomonas bacteria over here, you look at the plate, it looks kind of gooey, but it's actually bacterial ooze, they call it.
Or in the plant, when you cut the plant and open it, you see the ooze, that's the sign.
We have a picture. All right, pathogen signs, the fungal hyphae and spores, in this case powdery mildew. I used, when there was a big cut rose industry in this area, I used to study powdery mildew or rose. It's a very interesting pathogen. The sign, again, you have these overt signs of that powdery appearance. If you take on the leaf, if you look at it more closely, like with a 10x hand lens, you see this. But if you look at the pathogen at maybe 100x or so with a stereo microscope, you'll actually see the conidiaphore, which is the stalk that holds. You see the little conidia. These are these little single-cell conidia that are at the top. They're just ready to be blown off with the next wind gust. Okay, here another sign. The sclerotia, actually in this case Sclerotinia spiratorium, the mass of the mycelium that can be seen, it's all white. And then the sclerotia, and the sclerotia are these structures, the survival structures that kind of, mycelium that's kind of masked up and produces it's almost like a little black seed. These are visible to the naked eye when they're formed. It's usually later in the development of the disease cycle, though. You probably already have a dead plant by the time that you see these, unfortunately.
But the important thing is that because they remain in the soil, then next cycle, you're good.
This is the survival structure. So it's able to survive actually many years in the soil. So you've got to get rid of the spore if you're going to get rid of this disease out of the soil. And this is the bacterial exudate. In the case of Erwinia, that's attacked the internal portions of the stem. And when you cut into that, you can pull this out, and then the bacterial ooze that's produced by the bacteria can actually string out and like a sticky string that you see there. And then finally the nematodes, this would be a sign, these in this case, pathogen. The pathogen is a nematode, and these are these microscopic, non-segmented roundworms. If it's plant parasitic, you'll see a stylet in its mouth.